So give you guys a few extra points for being early. The first unofficial word for attendance will be bonus. You'll just get a little bit more extra credit than everyone else. Unless, of course, you're watching the video at a later date, and in which case, bonus is just your first regular old word. Bonus. See, Castro is online. Excuse me, Paula. As always, don't forget to email me with the words at the end. All right, let's see. Boggs is here. Harrison's here. And red. James is here. Esterson's here in red. Is that red?
Yeah, it's kind of dark.
Let me see. Wilder is here. Yeah, so far. Palmer is online. King is online. Again, don't forget to send the words before 9 a.m. Okay. So a quick reminder, if you ever join online instead of in person, you need to be logged into your WVSU email account. Sometimes, like what just happened right now, if you try to log in and you're not in your WVSU email account, it'll say, hey, someone's trying to come in. And right now, since it's early, I heard it and can let you in. If if I didn't hear it, or if we had already started the lecture, I wouldn't hear it, and I wouldn't know to let you in. So make sure you guys are using your WVSU Google accounts. Vincent, sorry, I missed you. Didn't see that, sorry. Vincent is here in red. Let's see, 2 minutes, less than 2 minutes to do some reminders, announcements. So it's been a really busy week for me because you guys had midterms. One of my other courses finished their semester, so they have finals. Another one of my courses started this week, so this is the beginning of their semester. One of my other courses, the online version of your course, also just had midterms. And then 2 of my other courses are just chugging along as usual. So I've been busy with that. Had a federal grant due, some information due, and that seminar yesterday. So basically what I'm saying is I'm really behind. And I know I just posted this, at least on social media, but if you need me, you're better off meeting me online for office hours. Don't email and say, can we meet for, just come meet me on office hours. I'm doing emails in the order they were received, and I've got at least 300 emails that I need to do. So if you email me today, I still got to get through days of emails. And by the time I get to your email, it'll probably be too late. So if you need me, just drop in online. I'm going to try to stay on longer than usual today. I guess that's it. for announcements and reminders. All right, it's 8 o'clock. Let's do attendance. Key is not here. Boggs is here. Bonner is online. Clay is not here. Dawson is not here. Ellis is here. Paula's online. Green is not here. Harrison is here. Henson is here. James is here. Kesterson is here. King is online. Lewis is not here. Linda Q is not here. Morrison is not here. Grace is not here. Ward is not here. Woodcock is online. Wilder's here, and Parks is not here. Did I forget anybody that's here? If you're online, don't worry about it. Just send me your words. Let's try to get this done. It's Friday. I want to get your weekend started and get my time to get caught up started as well. So let's jump back into where we left off. We were talking about independent assortment, but it's kind of talking about it in a different context. The first time we learned about independent assortment was during the meiosis conversation. And that was just simply, again, just to remind you, if chromosomes looked like where they came from, it wouldn't just be, for example, all mom chromosomes on one side and dad, piece of garbage, and dad chromosomes on the other, right? It could be that way. It could be they all line up like that metaphase, or it's going to drive me crazy. They line up independent of each other, right? So there is no rhyme or reason. It's all independent. Meaning, using this same example, of course, chromosomes don't look like letters, but if they did, let's say you had a gene for something on this mom chromosome, maybe the gene for hair color, which is, again, I'm oversimplifying, that's not really how it works. And then you had a gene for the amount of hair, but that would happen to be from your dad, right? So those would be inherited separately because they're on separate chromosomes. Now, on this other example, if it would ever stay up there, you could technically have, that's going to drive me crazy. Anyway, you could technically have the gene for hair color from your mom and the gene for hair amount from your mom. Oh, why won't that stay? Anyway, it is possible that you could inherit two different genes or alleles, technically, right, from your mom or your dad, but they're independent of each other. So it's possible that it could happen, and it's possible that it wouldn't happen. Let me back out and see what this noise was. I don't see any requests or anything. Nobody texted. All right, I don't know what that noise was. Back at it. So yes, that is what independent assortment is, right? So again, what we learned previously was they don't just line up mom on one side, dad on the other. Now in this chapter, we're kind of saying what that means, or some implications of what that means is, again, if you have two different genes and they're on two different chromosomes, they're not necessarily inherited together. Just because you got your mom's gene for one thing doesn't mean you're going to get your mom's gene for the other. Unless, of course, they might happen to be on the same chromosome and they don't get crossed over. But we'll talk about that later. So we already gave an example of that. Let me back up one. Right, we talked about this, for example. If the gene for pea color and the gene for pea shape, whether it be wrinkly or smooth, was inherited on the same chromosome, then they would always be the same. That would mean that every time you got a yellow pea, it would always be smooth. And every time you got a green pea, it would always be wrinkly. But that's not the case because they're completely different genes on completely different chromosomes. So yes, you might get one chromosome from your mom or from the male, one from the female. Sometimes you might get both from the female. Sometimes you might get both from the male. That's why we have all these different phenotypes with a dihybrid cross. Because all these genes, these alleles, they're independent of each other. That is independent assortment. So let's give another example. This is from your old textbook, but I kept it. Here are some possible phenotypes and genotypes of Labrador retrievers. So if you like dogs, hopefully this interests you. So in this case, for labs, having a black coat is dominant to having a brown coat. Obviously, I'm not going to test you on that. So when you're studying, don't take notes. Like black is dominant to brown. I like dogs, but not enough to be testing you on. Anyway, likewise, being able to see is a dominant trait for Labradors, and being blind is a recessive trait. And those two genes have nothing to do with each other. They're completely on different chromosomes, right? So let's look at some of these possibilities. Oh yeah, before I even say that too, this kind of goes back, this would have been helpful if we did this before this week's lab.
But when you look at it, this dog is black and can see, right? So all we know about this dog is that it has two dominant phenotypes. We don't know its genotype. We know it's big B something, right? That could be big B little b, could be big B. We don't know, because either way, that's going to result in a black coat. The vision, right? It's in big N something. It's the blank. Big N something. Because we don't know what it could be. It could be big N, big N, little n. Either one, it has the dominant phenotype, right? So because it has the two dominant phenotypes in this case, we don't know the genotypes. This dog happens to have a dominant phenotype of hair color, but a recessive phenotype for vision. So once again, we still don't know the genotype for its hair color, but we do know the genotype for its recessive vision, right? If it's recessive, you have to be homozygous recessive to show that phenotype. Then here we go, we've got a chocolate dog that can see. So again, because it has that recessive hair color, we know its genotype. It has to be homozygous recessive. But because it has the dominant vision, again, we don't know if it's homozygous dominant or heterozygous. Another way of saying we don't know if it's big N or big N little n, right? Either way, it's going to have that normal vision. And then finally, we have the blind brown dog, which is two different recessive phenotypes. So we know for sure that it is homozygous recessive for the hair color and homozygous recessive for the vision. Any questions about that? And again, this is just an example, so you don't need to memorize anything about hair color or vision for Labrador Retrievers. All right, moving forward. All right, sorry, I'm just making some adjustments over here. While I'm making adjustments, the first word for attendance, the first official word for attendance will be black dog. Even more extra credit if you can tell me who wrote that song without looking it up. Of course, I just have to take your word for it that you didn't look it up. But anyway, so again, independent assortment, right? So obviously, hair color and vision must be on different chromosomes. Those genes, those alleles, are not being inherited together. They're being inherited separately. Otherwise, maybe all black dogs would have good vision and all brown dogs would have or would be blind, right? That's not how it works. So let's look at some possible matings here. And again, this is sort of like a different thing I just showed you earlier when I was showing you that dihybrid cross, like that really big Punnett square, when we were looking at two characters, which was P color and P shape. Now we're basically looking at the same thing, or we're looking at two characters again. But this time we're not looking at the dihybrid cross, but we're still looking at the same results. So if you were to mix two dogs, right, and both of them are heterozygous for both of those characters, right? So again, heterozygous for color, heterozygous for vision, mating with heterozygous for color versus heterozygous for vision. If you were to mate those two animals, I mean, I doubt they would have that many puppies, but if you just kept mating those two animals throughout their lifetime, this is the ratio that you should expect. You would get about 9 of them that have the homozygous, the dominant phenotypes, both dominant phenotypes. Then you would have about 3 that have one dominant phenotype, but recessive other phenotype. And again, in this case, 3 recessive colors, but dominant vision. And of course, you'd only have 1 that has both recessive phenotypes. All this to say, if anything, once again, I just want to reinforce the fact that these characters, these genes, these alleles are obviously on different chromosomes. They're obviously being inherited separately. Otherwise, you wouldn't have this kind of mix. And the other thing you definitely need to know for the exam, I don't know if I reinforced this early enough or earlier, but this is a ratio you should associate with a dihybrid cross. 9 to 3 to 3 to 1. So, and again, when we're looking at these heterozygotes in your brain, just kind of think about like the F1 generation when we talk about the pea plants. That's the F1 generation, right? Because you're mixing 2 heterozygotes. When you cross them, you can expect a 9 to 3 to 3 to 1 ratio. And, it's not always perfect, And again, when the Labrador Retriever has their first litter, I don't think they have, I don't know what the typical amount of puppies is, but I doubt it's that many, right? So obviously, you're not gonna have 9 puppies that are black that can see, 3 puppies that are blind that can see, black that are blind, 3 puppies that are brown that can see, and 1 that's brown and you can't see, right? You're not gonna have that many puppies. But again, if they were to keep having puppies, whatever the number is, and you just kept having them for the whole life, you could expect a ratio about like that. Might not always be perfect, because this is all a game of chance, right? Because again, in meiosis, those chromosomes line up in the middle of the cell, and then they do that independently of each other, and then they segregate, right? So it is a game of chance. You won't always get this exact ratio, but that's about the ratio you're looking for. For that matter, and I don't even think I said this earlier either when we were talking about it before, when we were talking about the monohybrid cross, and we were talking about peas, right? So you have purple flower peas mating with white flower peas, heterozygote purple flowers mating with each other. And I always said you would get the 3-to-1 phenotype ratio. And that's about what you should look for, right? But in real life, this is all a game of probability. So it's not always exactly those numbers, but that's about what you're looking for. Anyway, any questions about this slide?
In reality, are there dogs actually born blind like this in a litter?
I think so, but that's a good question. I never even thought about that because I'm not into dog. I like dogs, but I'm not that into them. So yeah, that's a good question. The question is, in my opinion, is this a true example or is this one of those oversimplified things that textbook authors do? That's a good independent work question. If you want to look that up, easy 1 point. Some of you have a big goose egg. And remember, I'm about to put that big goose egg into WVSU Online, so your grade's about to drop, so you don't get surprised at the end. But let's go ahead and working on your points. And one of the things you can do working on your points is, that's an easy one. Answer a question. You don't have to write a paper, right? I'm asking you right now. Is being blind and actually controlled by one gene for Labrador Retrievers? Like, is this true, basically? So you can just send me an email. You don't even need to make a paper. I looked it up, and according to this source, and then just put little quotation marks, give me the exact answer that they gave you, and that's it. 1 point. And then again, obviously, if you find it interesting, you can actually write a whole paper about it and get into it if you want to. But if you want to just answer the question, that's 1 point. And I've been giving you so many options. I don't know why you guys aren't taking me up on this. By now, you should have at least 25, I think, points, right? I haven't kept track, but so many things, I'm like, this shouldn't be the answer. 1 point. Anyway, good question. Now, let's also focus back on this. This is a little bit less important for the exam in the grand scheme of this course, so I'm about to do the next thing pretty quickly. But once again, like I said, because this example, it has 2 dominant phenotypes, we don't know the genotype, just like you guys, right? When you did those labs and you filled out that table, you had to write both possible phenotypes. Big D, the word or, and then big D, little d, right? You had to write both possible genotypes because we didn't know your genotype. So, that being said, let's talk about using a test cross to determine an unknown genotype. Yes, there might be one question about this on the exam. First of all, what is a test cross? A test cross is a mating between an individual of a dominant phenotype, like that black lab I just showed you, but an unknown genotype. So once again, we have a black lab. Maybe we found it, at the pound or the Humane Society, whatever you want to call it. We know it's black, therefore we know it has the dominant phenotype, but that means we don't know the genotype. We do know it's big B something. Obviously, it has to have one dominant allele, otherwise it wouldn't have the dominant phenotype. But we don't know the whole genotype. So we, again, for the test cross, we mate somebody who is unknown to an individual that is homozygous recessive, because we know their genotype. If they're homozygous recessive, we know that they have 2 recessive alleles. Therefore, we could make those 2 and figure out the genotype of this person or this individual with the dominant phenotype. So here we go. It's easier when you visualize it. Here we have this black dog. Again, we know the phenotype. It's a black dog. It's a dominant phenotype. We know it has at least one big B, but we don't know Is it going to be big B, or is it going to be big B, little b? We don't know, because either one of those is going to result in that black color. Meanwhile, since this dog is brown, we know for a fact that it has to be little b, little b. So, we could cross them, and here's what we would expect. If this dog is homozygous dominant, like it is right here, right, or as that is right here, then this is what you could expect, right? 100% of the offspring would come out black. Because no matter what, and I don't know why the old textbook did it like this. I'm going to try to draw this on here. Because you're not accustomed to that. Now, in this case, one block does do it. But you guys, your brains are probably used to seeing the way I taught you. So let's do it the way I taught you. It's like the old textbook just took the easy way out. So if you were to cross big B, and big B, little b, you would get all heterozygotes. Big B, little b, big B, little b. Right? There's no way around it. You guys see how that works? Unless, of course, there's a mutation, and we'll talk about that later. But no matter what, again, assuming they had 4 puppies, all 4 of those puppies would be black. Does that make sense? So then let's look at the alternative. What if this dog is heterozygous instead of homozygous dominant, right? So that's what we're looking at here. And once again, the old textbook decided to, I don't know, make it look different. So once again, let's do it the way you're used to seeing it. We have big B, little b. And then we have little b, little b. So again, if these dogs were to have 4 puppies, we would expect, mind you, expect, that doesn't mean it was 100% of the time happen, but what we would expect statistically from the rules of probability is we would have half of those puppies would be black and half of those puppies would be brown. Does that make sense to everybody? So again, if they all came out black, we would say, yep, that's probably that dad or mom, whichever, must have been homozygous dominant. If they were about half and half, then we would say, oh yeah, they must have been, the other one must have been heterozygous. There are limitations to this. Can anybody guess what the one limitation is? And once again, let me emphasize the fact that this is all probability. This is not always like this, right? Because we don't know how they're going to line up. The chances of you getting this sperm versus that egg, right? It's this sperm versus this sperm, or this sperm versus this, or this egg versus this egg, right? It's all a flip of the coin. You're either going to get this one or that one. So that being said, what's the flaw here? What is the limitation to what I'm telling you? All right, I'll put it this way. Kind of take a few steps back. Imagine if we kind of set this up again and we hadn't already talked about all this, and I said, all right, we don't know this dog's genotype, but we do know this dog's genotype. We crossed them, and 100% of the, and they had 3 puppies. Let's just say 3 puppies, because that's a number we haven't dealt with. They had 3 puppies and they were all black. What do we know what this dog's genotype is now? They had 3 puppies and they were all black. Do we know the phenotype? Or first of all, let me make it easier for you before I even ask that. If you had to guess, if they all came out black, all 3 came out black, and you had to guess, which one would you guess? Is it homozygous dominant or heterozygous? If you had to guess, they have 3 of them, they had 3 puppies, all 3 were black. Homozygous dominant, right? That'd be a good guess. But I guess now my follow-up question is, do you know that? Forget mutations. And what? Can you tell me why? And you're right, you don't know that. Can you tell me why you don't know that?
Because they could have more not.
I think if I'm understanding you correctly, we're on the same page. The point here is, this dog, if it's homozygous dominant, no way, they all have to be black, right? This dog, if it was heterozygous, then it's 50-50 chance, right? The offspring could be black or brown. So if it happened to be all 3 were black, like, rules of probability is must be homozygous dominant. But it is possible that you just had 3 of these offspring. It's possible that you just, that it is heterozygous and you just happen to have 3 puppies that were black. That being said, I'm not going to ask you because this is taking too much time, but if I had time, I would ask you. If you had this happen and all the dogs were brown, then you could say without doubt that this one must have been heterozygous, right? Because there's no way to have a brown puppy unless this one has a recessive allele to give. So your old textbook didn't emphasize that. I just wanted to make sure that you were aware of this. This is all probability, right? So you need to be aware of things like that, right? If it's a black dog as the result, or a lot of black dogs as the result, probably it was homozygous dominant, but not definitely. If there was any brown offspring, then without a doubt, again, other than mutations, then without a doubt, we know that this parent must have been heterozygous. And again, for you non-biology majors, in a sense, this is useless to you, right? You're not gonna be breeding dogs, well, who knows, you might be. But this kind of critical thinking and scientific thinking is not useless. This is the kind of thinking you need to be doing, right? And again, not necessarily with Labrador, the coat color, but this kind of thinking, where I'm like, all right, you put this together and this, 100% should or are going to be this. And when you put this together with this, then you should get 50-50, right? Should versus definitely do. These are the kind of things you should be thinking about. Anyway, any questions about this test cross? All right, I keep saying the rules of probability, so let's talk about it. And I've got bad news for you. There's going to be a little bit of math on this exam, and it will be fractions. So let's talk about it. The rules of probability. I'd like you to figure these are the simple rules of probability. I know at least one of you, Kesterson, is doing statistics, so these rules of probability should be easy for you because you guys are, all statistics is based on the basic rules of probability. But here we go. Genetic crosses obey the rules of probability, which are the same rules that apply when tossing coins or rolling dice or drawing cards, etc. Right? So if you draw a card out of a 52 card deck, you have a 1 in 52 chance of getting any particular card, right? The 7 of diamonds. You have a 1 in 52 chance of pulling a 7 of diamonds. When you roll the dice, let me think. Oh, are they 6-sided? Yeah. If you roll a 6-sided die, you have a 1 in 6 chance of getting any one of those particular numbers, right? The chances of rolling a 3, you have a 1 in 6 chance of doing that. The chances of rolling a 6, you have a 1 in 6 chance of doing that, right? Or, and this one's more relevant to what we're about to talk about, luckily, because this one's the easy one. When you toss a coin, you got a 50% chance it's going to land on heads, and a 50% chance it's going to land on tails. Now that one is more along the lines of what we're talking about. Because how many alleles for each gene do you have? Assuming, and again, we're really simplifying things, but based on what I've taught you, where there's a gene, and then they have alleles, how many alleles are there for each gene? Possible alleles, so far from what I've taught you. Like that black dog, what were the alleles for the hair color? I just said black dog. So for the Labradors, how many different alleles were there for the hair color?
2.
2, right? Either the black allele or the brown allele. So far, everything I've taught you has been either this or that. So if you're talking about somebody who's heterozygous, you have a 50% chance if you flip the coin of picking any particular sperm or egg out of their body and looking at it, if you could somehow look at their genome, and there'd be a 50% chance you would get one allele and a 50% chance you would get another. So with that in mind, let's take a look. This is kind of what I'm saying. So we don't even know what B is in this case. It doesn't matter. What we have is 2 heterozygotes mating, right? So this female, she has a big B, she has a little b. Those are her 2 different alleles. This male has the big B and has a little b. So those things, once she goes through meiosis, they're going to separate from each other. There's the female. There we go. They're going to separate from each other. So half of her eggs are going to have a big B, half of her eggs are going to have a little b. So if we could somehow look at this female's eggs and randomly pick them out of her body and look at the genome, 50% of them, like the chances of you picking one that was a big B is a 1 in 2 chance, a 50% chance. The chances of you picking one that was a little b is a 1 in 2 chance, a 50% chance, right? Or another way you could say that is, if you pulled out 100 of her eggs, by the rules of probability, you should expect about half of them to be big B and half of them to be little b. Does that make sense to everybody? About that, right? And again, flipping a coin, if you do that 100 times, you would expect it to be about 50 times heads, 50 times tails, but not necessarily, right? It could vary a little bit, but chances are, statistically, it should be half and half. Same thing with this male, right? You see there in his cells, you look at those chromosomes, he has 2 alleles for each, right? Because he's got a chromosome from his mom and a chromosome from his dad. When he goes through meiosis, those things segregate. So now 50% of his sperm have a big B, 50% of them have a little b. So the chances of you picking out one particular sperm and finding a big B is 1 in 2. The chances of you picking out a sperm and picking out a little b is 1 in 2. And again, using the same thing I said here, maybe I'll change the numbers up. If you were to look at 1,000 of his sperm, then you would expect about 500 of them to be big B and about 500 of them to be little b. Everybody with me so far, right? It's either this or that. It's a flip of the coin, and that's how that works. So therefore, basically, we're looking at what you've already done with these Punnett squares, except now we're just putting math behind it, right? So there's a 1 in 4 chance that you have a big B, big B. There's a 1 in 4 chance that you'll have, did I say big B, big B? Yeah. There's a 1 in 4 chance you have a big B, big B. There's a 1 in 4 chance you have a big B, little b. There's another 1 in 4 chance you have a big B, little b. And there's a 1 in 4 chance that you'll have a little b, little b. Now, where this comes in mathematically is, again, so when you look at this whole thing, and this is from your old textbook, I don't know why they didn't do this. So these are the same, right? Big B, little b. So in this case, you would add them together. So if I were to say heterozygous female for B mates with a heterozygous male with B, and again, you would need to know heterozygous means 1 dominant, 1 recessive, 1 dominant, 1 recessive. What are the chances of them having an offspring that is also heterozygous? So you would do the Punnett square, and you would say, well, you can either do the math or you can just look at it. Looking at it's a lot easier. So let me just ask you before I do the math. How many, let's again assume they had 4 offspring. How many of these 4 offspring are heterozygous? And this is exactly what you guys did in lab. You didn't necessarily do the math, you just counted it. How many of them are heterozygous?
2.
2, right? Yes. So there's a 2 to 4 chance, right? And that also, reduces down to a 1 in 2 chance. Does that make sense? All right. And then mathematically, again, to me, in this case, it's easier to just look and say, oh yeah, 2 to 4. It's right there. But if you do that mathematically, if you have the probability of one thing happening and you're asking what are the chances of this happening or this happening, then you add them together. So 1/4 plus 1/4 equals 2/4, right? And that's where we got to, 2/4, which of course reduces down to 1/2. So that's how that's going to work. And it's going to be basic math with some fractions. So I might say something like, all right, maybe I can do that example later. I'm trying to think of how I'm going to say this. All right, maybe again, let's say I give you this same example that we see here, heterozygous female mating with a heterozygous male. What if I said, what are the chances of that woman and that male having a baby that is, well, first of all, I'll ask you this and then I'll add that other part. What are the chances of these 2 people having a baby that is homozygous dominant? So just look at the board, you don't even need to do math, just look at it. If they have 4 babies, what are the chances of one of them being homozygous dominant?
1/4.
Yeah, well, yeah, 1/4, right? Good. Now here's where it gets as tricky as it's gonna get. What are the chances of them having a boy who's homozygous dominant as opposed to a girl? That's a little bit trickier, and this is where I'm gonna teach you some math. Like he already said, the chances of having a homozygous dominant, because I just randomly chose that example, could have been any of them, and might have different numbers. The chances of having homozygous dominant, as he correctly said, is 1 in 4. What are the chances of having a boy versus a girl? Don't overthink it. Forget the homozygous dominant thing for now. Just imagine this is your two of your friends, a guy and a girl that you know, and they're like, hey, I'm pregnant. What are the chances of them having a boy?
1 to 2.
Good, 1 and 2. Perfect, 1 and 2. So here's the trick. This is as tricky as it's going to get. When you're talking about two things happening together, then you have to multiply the probabilities. So the chances of having a homozygous dominant is 1 in 4. Chances of having a boy is 1 in 2. So the chances of having a boy that is homozygous dominant, 1 in 8. Make sure everybody understands that. Well, I shouldn't make sure that, because again, I'm not going to ask that many questions. You might have 1 or 2 questions like this. So if you get stressed about this, don't worry about it. Well, come to me for help, but then also don't worry about it because it's not going to affect your grade too much. So when you talk about the probabilities of two different things happening, you multiply the two to figure out the entire probability. I'll give you another example, another non-biology example, since I've already said it. What are the chances of rolling a 6-sided die and it landing on the number 3? 1 in 6, good. Now, what are the chances, completely separate, let's not even talk about, let's forget, we already talked about that, let's talk about something else. What are the chances of, I don't know, pulling out a Queen of Hearts out of a 52 card deck?
1 in 52.
1 in 52, correct. Now, if I said, what are the chances of both of these things happening? Like you roll whatever number I said, and you pull out a Queen of Hearts, what are the chances of both of those things happening? You would multiply them. And, I'm good with math as in algebra because I'm really good at manipulating variables, but that good at multiplication in my head. So let's take a look. 6 times 52, so that gives us 312. So there you go. And again, I don't know why I'm spending so much time on this because it's not that complicated. And like I said, if there are any questions about it, there'll be only 1 or 2, so it's not going to make a big difference to your grade. Plus, once we're done with this chapter, we're never going to use this math again. The concept is going to be there. It'll be helpful to understand evolution and things like that. But the math, once we're done with this, we're done with it. So anyway, any questions about that math? That's another independent work thing you can do. I think this might be the first time I've suggested this, just for fun. I don't even know how I would grade it, but if you just wanted to demonstrate that you understand the rules of probability, just write a few scenarios like, what are the chances? Chances of this happening and this happening would be this, and show your math. And the chances of this happening or this happening would be this, and show your math. Oh yeah, sorry. Before we move forward, let me give you one more example, because I keep saying the chances of this and this happening, right? Or the chances of this and this. Let me give you one more example of having this or this. So again, what are the chances of having a boy versus a girl? You guys have already said it.
1 to 2.
1 and 2, right? So what are the chances of having a girl or a boy instead of a boy or a girl? What are the chances of having a girl instead?
1 to 2.
Good, 1 and 2. So, and this is all like words here, but what are the chances of, instead of saying, let me say this carefully. In that case, again, if a couple's pregnant and they say, hey, we're pregnant, what are the chances of them having a boy or a girl? So mathematically, instead of this and this happening, we're saying this or this happening. So instead of multiplying it, you add it, and that gives you 2 and 2, which comes out to 1, or basically 100%, right? The chance of having a boy or a girl, yeah, that's your only 2 possibilities. Yeah, it's 100% chance. Just like flipping a coin. What are the chances of having a heads or a tail? As far as we're concerned mathematically, 100%. Of course, there's that really small chance that it could land like this and somehow perfectly balanced, but that's a whole different story. Anyway, are there any questions about these rules of probability? All right, so the next word for attendance will be poker, as we're talking about cards. Poker. All right, let's talk about family pedigrees. And this is also, again, from your old textbook. And I don't remember from the old textbook. I'm not even sure if this is in the new textbook, but I want to talk about it because this is probably a little more relevant to your everyday life than some of the other stuff we talk about. Mendel's principles apply to the inheritance of a lot of human traits. So, right, all this work he did with pea plants, it's not like that only pertained to pea plants, otherwise he wouldn't be famous. Like, peas are cool, I guess, but they're not that cool. Like, people don't care that much about peas. So what we're saying here is that a lot of the stuff that I just talked to you does apply to a lot of human traits. So the next slide I'm going to show you illustrates some alternative forms of some of the stuff that we've even talked about in the lab. Widow's peak, as you know from lab, not that you need to memorize this, but remember I didn't memorize a lot of these things. We've all been doing this for so many years. Widow's peak is dominant, no widow's peak is recessive. Free hanging earlobe is dominant, connected earlobe is recessive. One that we didn't do, freckles are dominant, no freckles is recessive, right? So these are some of those examples that follow these rules of probability. The rules that Mendel figured out, these are some of the examples that follow those rules. And I keep saying that because as you're gonna learn later, there's those exceptions to the rules, but for now, let's just focus on the rules. So let me ask you this. How many of you, look, we see here, no freckles are recessive. How many of you know more people that have freckles versus don't have freckles? Because I personally know more people without freckles than with freckles. How about you? Does anybody know more people with freckles than without freckles? Just curious, no? Okay. And as we demonstrated in this lab, this last lab, some of these characters that we looked at, like more people in the class were dominant, but then some of them, more of us were recessive, right? So that brings us to a term you need to know, which is called the wild type. The wild type trait is the one that is most seen in nature. And it's not necessarily specified by dominant allele. So again, just because it's dominant, that doesn't mean it's more prevalent in the world. It just means when a dominant allele is with a recessive allele, it will show up and the recessive one won't. But that doesn't mean it's more common. So again, freckles is a great example. Right? As we just said, we all know more people without freckles than with freckles, even though the freckles are a dominant trait. So make sure you know what a wild type trait is. That being said, let's jump into this family pedigree. How can human genetics be studied? Right? We can't do what Mendel did with peas. Right? Think about because remember what he did with peas was he took this plant and he ensured that it mated with this plant. Right? You can't do that with humans. It's not ethically. You can't say, I really love to see what happens when you mate with you. You guys go have kids, right? You can't do that. That's not ethical. So instead, geneticists analyze results of matings that have already occurred. Because again, for humans, it's unethical to have people, mate for the sake of science. But people do mate all the time to the point where we have over 3 billion people on Earth. So we do have some data to deal with, like some subjects to look at. Therefore, we can then assemble this information into something called a family tree. I'm sure you're all well aware of what a family tree is. We can also call that a pedigree, at least in this context. Usually when people think of pedigrees, they think of purebred dogs or purebred horses or something like that. But they can also be used in human genetics. So these pedigrees can be used to determine if a trait is sex-linked or not. And again, this is from your textbook. It can determine a lot of things. So I kind of want to put an X or a circle. I don't know. I need to fix that bullet point. That kind of implies that that's all that it can be used to determine. It can be used to determine a lot of things. But in addition, it can also be used to determine something is sex-linked. So let's give some examples. And it's funny too, because once again, this next example, the next bullet point, is free versus attached earlobes, and that is not a sex-linked thing. So we'll talk about sex-linked later. Right now, let's just talk about a pedigree using the earlobes thing, which is a good example because we've talked about earlobes. So here we go. Squares are males, circles are females. If it's colored in, that means they have the dominant phenotype, which is attached earlobes, right? And if it's white, we know they have the recessive phenotype, which is, I'm sorry, I said that wrong. Forgive me. As we know, as I should know, and again, I can't remember because it's not important. If I remember correctly, the free hanging earlobe is the dominant. Yep, just wanted to double check. All right, free hanging earlobe is dominant. So if it's a white colored box, that means they have the dominant phenotype. If it's shaded in, that means they have the recessive phenotype. So remember, there's three different types of genotypes we're looking at. We're done with the phenotypes. We can see the phenotypes. Dominant, recessive, dominant, recessive, right? That's easy to determine. What we don't know is the phenotypes. genotypes. So remember, there's 3 different possible genotypes. It's homozygous dominant, heterozygous, and homozygous recessive. For some of these, we don't know what they are. For some of them, we do. Which one of these do we automatically know what their phenotype or the genotype is? And don't overthink it. Like what one of those 3 genotypes, we automatically know when someone has that genotype. Think about the labs, or the Labradors, and also the lab we did this week. All right, no one wants to guess? I'll give it to you. Obviously, if they have the recessive phenotype, they have to be homozygous recessive. Make sense? All right, good. That means we know that every single one of these shaded in is little e, right? We know that. They have to be homozygous recessive. Otherwise, they wouldn't show the recessive phenotype. So we go ahead and fill those in. You might get a question like this on the exam, or it might be like a after the exam extra credit thing, because obviously it's going to require you to fill some things out, figure some stuff out. All right, so there we go. Now think about Labradors. What we do know about these other ones is at the very least they have one big E, right? We know with big E something. Everybody with me on that? I hope that makes sense. If they have the dominant phenotype, obviously they have to have at least one dominant allele. Okay, getting there. This might be the last thing we talk about for the day, is this pedigree. So the quicker we get this pedigree done, the quicker you guys go enjoy your weekend and I can get caught up and graded. All right, so now we have to figure out what these genotypes are. I mean, technically we could start anywhere. I'm just starting with that because for me, I know this one seems easier. And I can tell by looking, I know what these genotypes have to be. Does anybody else know? Well, do I want to say it that way? Well, let's look at the possibilities. Just like we did with the Labrador, all right? What if it was big E, little e? What if they were both heterozygous? Let's see what would happen if they were both heterozygous. Then we would have one of the offspring would be big E. Big E, should have made that bigger. Or maybe just draw it on the board. That's what I'll do. No, they won't be able to see it online. Big E, little e. Big E, little e, or little e, little e. All right, so we know based on this genotype, or this, based on this that I just did, we know that this is a possibility. They could be both heterozygous because that would end up with homozygous recessive, which we have. So let's look at another situation. And maybe this one I can do on the board. All right, let's look at big E versus big E, big E. Okay, what if they're both homozygous dominant? I'm not even gonna waste my time doing that Punnett square, because hopefully, as you can see, 100% of their offspring would be big E, right? You can see how that's all we could get from that, right? And we know that's not the case because they have some homozygous recessive offspring. Now, what if they were, one of them was big E, and the other one was big E, little e? Let's look at those possibilities. You should get about 50% that are homozygous dominant and 50% that are heterozygous. Once again, that can't be an option either because none of their offspring would be little e. Therefore, we know, because I've done all the possibilities, that the only one would work would be the first one, which is up there, right? Two heterozygous go together. It has to be two heterozygotes. It would be the only way that you can have two homozygous recessive. There we go. We figured that out. But what we don't know then, based on that, is, so again, we know this has to be the mating. We know they have to be heterozygous. So we don't know if these offspring are big E, or if they're big E, little. We still don't know that. We have at least figured these genotypes out, but we haven't figured out these, and there's no way to figure this one out, but there is a way to figure this one out. Once again, since we already did the math and already showed you, I'm not going to do it again, but we know for two dominant phenotypes to have a homozygous recessive offspring, they both have to be heterozygous. We know that because to be homozygous recessive, you have to have two recessive alleles. Therefore, you have to get a recessive allele from your mom and you have to get a recessive allele from your dad. Therefore, they both have to be heterozygous. Once again, we can't really decipher that genotype. And then here, well, let's just leave it at that. So you guys see what I'm saying, right? You get the gist of what I'm saying. So you might have to do something like that again, maybe as extra credit. Or maybe I can find a bunch and give them to you, maybe for independent work. Nah, because you need to be writing for independent work. We'll just call it extra credit. Anyway, the last word for attendance is going to be ear. And then we're gonna leave, we're gonna end 5 minutes early. And again, if you need me, I'm going directly downstairs, I'm hopping online. I've got a lot of catching up to do, so if you need me, meet me online for office hours. That way you're skipping the line of emails and I can help you immediately. And hopefully somebody else will come in and then with your permission, they can sit in on me helping you and they can learn from you needing help. But if not, you can let me know and say, hey, I wanted this to be one-on-one. In which case, we will do that.